hear me? Now it's okay?
Hello. Is loudly enough, I think. Okay, okay. So a lot of people still didn't come yet, so check it. Can anyone talk to me a little bit? As usual, feel lonely in here. And it's good if you turn on your camera a little bit. Can you? Nice. Thank you. Okay. So that's good to see you again. Not, it's maybe more than half of the class is going somewhere. So how are you? Are you still okay, right?
Yeah.
Any kind of expect from the pandemic already to you? Some change from your side?
Three more kilos from the test holidays.
Okay, so it's maybe it's common effect for anybody. So it's good to see you, and thank you very much for joining with me. So this week we will continue to some chapter of the book and also the course. And last week, okay. So in last week we already discussed quite a lot about the autosomal inheritance, the autosomal, with the strictly kind of follow the theory of the Mendel experiment and also his rule, right? So in here is today we also the same thing. We also follow a little bit, also follow the Mendel theory. It's quite of strictly, and sometime in the end of the chapter we kind of jump out a little bit his theory a little bit. Different with very big difference between this week and last week is last week we talked about the autosome from the chromosome number 1 to chromosome number 22. Today we will focus on just a very limited amounts of the number of chromosome. We just talk is sex chromosome is X and Y in humans. So but it kind of small number, but and small gene also, but it's quite complicated compared with the last week. So so at the first, I kind of want to discuss a little bit. I want to discuss a little bit about the science of the medical genetic a little bit, because I also involved this one is a long time already. It's not really just in human, but quite of the scenario and the principle is quite of the very close with genetics. So you see that you learn a lot about genetic, and in maybe cell biology you also have talking about genetics, and also you have also in the genetic class. Is we learn a lot of genetics, and this is we use the genetic for human disease understanding, and also try to maybe fix this, cure the disease. But for when you can read the book and when you think more about that and how you're feeling about this science, you see this science is kind of not very clear science. You see, right? Something you cannot see in your eyes. You have to use some kind of the method like molecular technology to check it. And a lot of the theory is that we cannot check it yet. And I also have a thing, but it's not confused. Thing is more and more clear by the work very hardly of the scientists. And also the technology is more improved and is more evident for the theory. And of course, some not exact, inaccurate the theory, we also remove it already. But in the end, is no, not in the end. Yes. So this science still is not very clear. The science, you can see that like different with math, and 1 + 1 you have 2, right? And this is you're not sure. So last week we talking about the case of the incomplete penetrate, right? So that means the one had the mutation, but they didn't have the disease at all, or some kind of variable in the variation in the expression. That means the one had the mutation, and somebody has the same genotype, but they express different with the incomplete penetrate. And many things affect to the outcome, the phenotype outside. So that is like I also thinking about this. Like the thing is not clear. It's quite mysterious, and also it's beautiful, right? A little beautiful. Like what? For example, the Mona Lisa picture. So we don't know if she smile or she angry at all, right? And we're not clear. We're not clear about the picture of the Leonardo da Vinci. So that is make this picture is kind of one of the most expensive picture. Which one is beautiful? Which one the mystery is quite expensive. So this science also is quite expensive to take care, because if you are not have any kind of thing related with the disease, so you don't care. But if the one people is have the disease case, they really care, and they spend a lot of their effort for fixing the disease. And somehow, if they are the carrier, and in the family they have the related people have the symptom or the phenotype of some kind of genetic disease. So we should care about this case. So I'm worried because I also maybe have that gene, the bad gene in my body. I mean, I can transfer to my son and my daughter. So I also very quite worried. So that is somehow you feel is not clear, and your conclusion may be not correct. But when you learn more, and actually is the scientific ICUCs, we always try to make clear about that disease, and that's very important for human. So some of thing is because we talking about Mendelian disease, and Mendelian disease relate with mutation on the chromosome and the transmission pattern. And you see that mutation is quite rare, right? Maybe just some hundred, some kind of 10,000 or 100,000 people, and to have one people. Number of people affected with disease is quite small. So we also is maybe you put a lot of money for investigation. Maybe you have no any kind of feedback. Like you lost a lot of money, but just treat for some people maybe it's not fair with you. But for science, it's like good because we more and more understand about the disease in the single gene, and we will develop our ability to understand at a lot more complex the disease, this complex etiology of disease. So today is, last week we are a little similar with Mendel, and today we will talking a more special case is also interesting, is sex link, and something we will talk out of the Mendel rule is a non-traditional mode of inheritance. It's related with something like imprinting gene. It's not clear yet. Some is weak, and we're talking about the mitochondria genome mutation, and also the consequence of that mutation. So I just take this slide from last presentation. So we had two cases, like we had a multiple gene disease. So this one, we after midterm, there one chapter we focus on discuss about multiple gene disease, and in here is we just focus on single gene disease. So this is autoimmune system, and what I want to concern you is today we will talk about the X and Y and mitochondria genetic material mutation making the disease. So in here is the sex chromosome is quite not small, because of, you see that from the chromosome 1 to chromosome 22 is that the size is decrease, right? Chromosome number 1 is the largest chromosome in our cells, but X chromosome is not small chromosome, because it's not, they have some gene related with the sex development in development, and Y also same thing. But X and Y is a very special chromosome. So in here, X chromosome actually they are big chromosome in human and also in animal. So this is the size of the chromosome, 155 million base pair, quite big, and it costs for account for 5% of the genetic material in the nucleus. And the Y chromosome is kind of very small, and it's just 60 megabytes, 60 million base pair. But 60 million, but number of gene in the Y chromosome really small, about just very less than this number, 63. But in X chromosome is a lot of gene. Here we have more than up to now we have 1,300 more gene is characterized in X chromosome. That means X is much more bigger compared with the Y chromosome in gene number. And the mitochondria, this one not correct, sorry. This one is very small mitochondria. But mitochondria is small, but also have a lot of gene there. Why is a lot of gene there? Because of the mitochondria, you know, is not original from the human, right? It's maybe from the bacteria with ancestor cells acquired in the development. So it's quite a small, same size with the plasmid in the bacteria. So it's small, and the thing is they have no intron. So a lot of gene in this plasmid. We have in the end of the presentation, we will talk in more detail about mitochondrial genome. And even small, but mitochondria also coding for very important gene in the cells, because this is powerhouse of the cell. So without, if the engine of our cell has some problem, we really have some problem, because we have no energy, and energy is the most important for the activity of the cells. So so today we will talk about some material. We had a mutation in the X and Y and mitochondria mutation related. And the more important is the transmission pattern compared with sex chromosome link disease together with different with their autosomal chromosome. But before we go deeper about the sex chromosome mutation and inheritance pattern, we have to remind about the very important activity. I mean, important process in the normal cells, especially in the women cells, is the X chromosome inactivation. We call it X inactivation. So you see is that is in the autosome. So we can have the any kind of gene have the double dose because we have the two alleles, the two set of that gene, one from mom and one from dad. Except for some thing related with the except for some all the gene related with the sex chromosome. You see, it's in the man. In men, we have just one X chromosome, and Y is actually Y have very large gene in there, and the amounts of the protein in the man is quite up compared with the autosomic just 50%, right? Because it is just a one dose. But in women, we have the two X chromosome. That means it's similar with, similar with the autosome, but actually is the double compared with the man. So that is double compared with man is not allowed in the normal development of the female. So one of the couple X chromosome will be inactivated. So it's for what? For the dose compensation in sex chromosome protein. So the dose from the 100% should be decreased to 50%. Same in men should ensure for normal development of the women's cells. So this one is a state in the hypothesis by Lyons in the 1960. So she don't have any kind of evidence about the cell yet, but she just sees some kind of a picture in their microscope about the cells. So she has stated, so we have the early random activation in X chromosome, in one X chromosome, X female cells. So I mean is, as you all know, right? So we have the inactivation of one X chromosome. Just say only one X chromosome is work, and another chromosome become the bar body, right? That process is kind of random. I mean, it's why is random? Random because of in the beginning, in the zygotes, we have in the women's cells, we have the one X chromosome from father and one X chromosome from mom. So the random is the cell can randomly select for which one they don't care. So from when the cell propagates, and in next cells they can check that cells, and then the father X chromosome may be inactivated, and another cell is maybe the mom mom X chromosome is inactivated. But that's random, just but early stage. But after that, it's become a fixed process, and the cells, the chromosome when they will inactivate, and they always inactivate. That chromosome is still inactivate to the, I mean, if that cell is the mom is the X chromosome from mom is inactivate, and the daughter of that cell is always the mom. The chromosome is always inactivate. That's a fixed process from when they choose already. But when they are But I want to say something. But how about when the germline cells, when the germline cells, when the germline cell making the when the germline cell making the what? When the germline cell making the gamete. So how about the bar body? Can you answer? They still inactivate, or they how about they have some kind of the mechanism to reactivate the X chromosome in the gamete making process. You understand the question? Yes, I mean that you see that the X chromosome in any women cells is the one X chromosome is inactivated, right? Become a bar body. So how about in the germline cells making a gamete, making the X? How about that's bar body? So they can revert to the normal chromosome, active, or still keep the inactivated state. Get it? Oh, great. Very diverse in the answer. But the thing is, we have to reactivate again. So we have some mechanism. Because you remember is that's quite easy to also is we just need some protein to reactivate the gamete, and also some you know is the kind of decondense, right? No, condensation of the plasmid region, auto hetero. So we just remove some mark of the histone tail acetylation, and also we also remove the methylation in the DNA, so we can reverse the activity. Okay, back one slide, I forgot to add one also very important question. This one is, I think when I prepare the slide, you see the sex chromosome is quite big and very important, right? Very important for the cells. And you see is in the autosome. You see that in the autosome, we have some the crossover. I mean, the recombinant scene is happen a lot. I mean, it's the evolution. We had a lot of mutation about there, a lot of mutation related with recontract the plasmid between the species and species. I mean, and also is you can see the result that means between the species, the chromosome structure it changed a lot, even though the chromosome number, right? Chromosome number. So, but the sex chromosome is quite very important, and then they maintain some gene very important for the cells. So I had a question about how conserved. I mean, the structure of the sex chromosome is stable or not among the animal species. This is related with the evolution. You can think about bigger picture. And I mean, it's also same meaning question. So they are involved independently. I mean, it's not related with another chromosome, because our chromosome may be mixed together, but this chromosome is not. Maybe not. So you accept that, or and can you suggest some mechanism related with your suggestion? You get the question? Hello.
Sorry, could you please repeat the question?
Sure. So you say that is sorry. It's quite complicated, not very complicated question. So I mean, it's a how conserve. I mean, it's genetic conservation. You know it, right? The genetic structure of the X chromosome among species to species is different or not, compared with the change of the normal chromosome. I can say the normal chromosome, I mean, it's autosome. Between species, it's very different, even the number of chromosomes, right? But sex chromosome, we have just only 2 chromosomes, and I ask is they conserve or not. You know, what is genetic conservation, right? Another? Thank you, Khoa. Okay, great. So you see that because sex chromosome, I also search myself. I search myself because this is not my research. It's quite big. So a lot of publications say already they conserve because sex chromosome is sex chromosome. Yeah, we says yes, right, it's correct. It still have the recombination, but it's conserved compared with autosome because sex chromosome is why very important. If we have some problem in there, the disease will be expressed, and then the animal will die. Absolutely. Maybe some gene jump in there, and the sex chromosome effect will be, you will see today, effects of the chromosome as sex chromosome. So it's conserved. But why is conserved? The mechanism, I didn't read that yet, but I think it's about because sex chromosome and X and Y chromosome are quite different in the size and also different in the sequence. So they don't have some mechanism is we have in autosome recombinase, right? Recombination. So recombination, we need the homologous region. The homologous region, so the enzyme can mix that, and then this jump to this. I mean, it's from this segment of chromosome jump from this chromosome to another chromosome because of homogeneous. So in the sex chromosome, maybe we don't have this. This is one mechanism I can think about. But if you have time, you just have to study about this. But also very interesting, and a big paper also submit this one. Last time I think about 3 years ago paper, and I go to Nature about this topic. So because of in the X chromosome inactivation, talking about which original mom or dad X chromosome. So we have the female is a mosaic, right? So 50% of cell is a father X chromosome inactivate, and the 50% mother X chromosome is inactivate. 50 and 50%, that is in theory because of random, right? Like the tossing the coin. and the female is mosaic. So and male, how about the male? Male is we have just only one X chromosome, and the Y chromosome didn't contribute is not too much in the development. It's just in the sex characteristics of man gene in there. So male is we call it hemizygous. Hemi is half. And normally we call homozygote. Homozygote is both. We have both, and they're similar, but hemi we have just half. Okay. So this is the figure illustrate for the female cells. So the female's body is if we cut, no, don't cut. If we check in any kind of the part of the female, so we have the 50%, and they are knit together, become the quite a different in genetic, because of the bar body is can be the can with the can be the mom or dad X chromosome transfer. So thing is, I also had a question myself. So if there and you also have to ask yourselves. So if the disease gene, for example, in the father X chromosome, so that means they also express in the cell, right? They also express in the cells. They they different with the autosome. Autosome in normal cells. So autosome is that cells the concentration of chromosome. If we have the heterozygote, and this also is the same with heterozygote. In the autosome, if we have the heterozygote, the amounts of protein may be decreased 50%. But if you have the, for example, you have the in the female cells, we have the father is a have mutant in X chromosome, and the mother is already inactivated. That means in that cell is no protein making at all. The concentration of protein is 0, right? Accepted, right? So and mom is a reverse way. So but we have the 50% cell is okay and 50% is not okay. That maybe is okay for women. I don't talk about the talking the I mean the relationship between the next two cells, but at least 50% in women's cell is normal, and 50% of the women's cell is abnormal. So maybe somehow they can rescue the side effect, but kind of the manner is different with the autosome heterozygous. Okay, this is I come back with some science, some facts related with the cell's biology. So you remember this one, right? The cell memory maintained by epigenetic mechanism. So in here is the first we have some mechanism related with. First is the methylation of DNA. So methylation of DNA related with adding the methine group CH3 in the C next to the G in the downside. We have that is CG, the CG base pair like this. So some enzyme we come and recognize this site and add the CH3 to C side C. And when they break this, because of a complementary propagation of DNA, so we have this one is a one breaking between two, and new strain is new strain. They don't have the methylation pattern, right? Methylation don't have the new DNA adding it. They don't have it, but some enzyme will be come and based on the methylation pattern of the old strain, and then they propagate the signal of the CH3 in the new strain. So finally, we have the memory of the methylation system. This is make sure that the cells already differentiate to the specific cell. Maybe the lung cell is still be lung cells and not become the very different cell in that population. And the same thing for histone histone modification. So this most related is the most effect modification is acetylation. So we need to remove or add an acetylene for keeping the cell mark to making the same some kind of a function and the roles of the cells. So that is some mechanism related with the memory of the cells. So this visualize for this one. So if here is the maternal is so this is paternal cells is inactivate, and this is also daughter cell also the same thing. The paternal is also inactivate in the same mechanism like this way. So this one also you may see already. So we have the one example is a calico cat. So we have the in normal is a male and a female. So we have that kind of relay with a different color, but in the female because of in the female cat because of the random inactivation of the X chromosome. So we have the calico cat with the different size in the body with different color like this. And we also have the one example in the human that is related with albinism, ocular albinism in related with one gene related with X chromosome. So we did also have this one in the last presentation, right? So the gene related with producing melanin is also have mutation. So this related with X chromosome, and you can see it's the mom. So we have seen the color in the ocular is kind of it's different color, is part patch like patch. So some is very dark and some kind of light in here. That is a kind of intersect together, and mom, you see that because of C have there two X chromosome. So effect is kind of it's okay, but in son, when she have the son, so the son is only have the one X chromosome, and the disease is kind of heavy expressed. So the pigment in the eyes kind of very less in this case. So you can see that it's kind of, the kind of less color is a lot in the ocular. Oh. Okay. So I also checked, and we have a new finding for X inactivation. So it's not early, very early, but kind of early. It start X inactivation is start about 7 to 10 days after fertilization. So in that time we have the dozen of cells, but not much. The inner cell mass is kind of some dozen of cells only. And inactivated, we start at about we have the X inactivation center. That means that reason is about 1 megabase, 1 million base pair, and expand to another size. I mean, we have X chromosome is a 155 base pair, 55 megabase, right? And one base pair is somehow they have some gene in there, and recruit some kind of protein come to start the inactivation. This should be the inactivation center, and then they expand to another size of another size of the X chromosome. Oh, sorry. Okay, now.
Yeah, it's okay.
Somehow I stopped sharing. Okay. So but in the placenta, we have just only the inactivation in the X chromosome from male only. So placenta is quite important, and we need a female. Somehow this is in the mechanism to make the placenta. So only the X chromosome from male is inactivated. And this one we crossed already. So this is we have the reactivation in the female germline stem cell for making a germline. And the bar body number is only one less than number of X chromosome. That's kind of confused, right? For example, the women, somehow she have about maybe 6 X chromosome. Somehow it's happened. Somehow it's happened in real life. Somehow she have the 6 X chromosome, XX chromosome. And this sentence say that 5 chromosome will be inactivated, and then they let only 1 chromosome active. So to balance the normal dose of the protein. And but this also make up one question. So I mean, is in the glyphanther, right? Glyphanther. So in the glyphanther, we have the XX and Y chromosome. So it means in here, XY is the man, right? So XY is normal, but we have additional X chromosome, and this one should be inactivated, like this sentence, right? So the bar body number is only 1 less than number of X chromosome. So in here we have the X chromosome, 2 X chromosome, and 1 X chromosome should be inactivated, and that's true for glyphanther people. But why the disease phenotype also expressed? Why is this is also disease form? Because of if the X chromosome is completely inactivated, so that's normal dose of protein, right? So the answer is the inactivation is not incomplete. It's an incomplete process. That means some kind of reason in the X chromosome is not inactivated, especially in the tails of the chromosome, in the near the telomere region of the X chromosome, we have no activation. I mean, 50% of gene in the X chromosome is escaped out of the inactivation. Sorry, this one is super inactivation. Question? Mm-hmm. Yes. Yes, in the previous slide, we say that it's the random process, and this random is not maintained in the placenta, and only male is inactivated. Okay. So is why is we see that is if we have the additional X chromosome, the cell is inhibit the dose and keep its normal dose of protein on the X chromosome by the inactivate X chromosome. So like if you had a question like if you have the 6th chromosome, and how many bar body in there, you can say like 5 bar body in the cells, because of they just let about 1 X chromosome active. But this also not sure about the disease thing because they also develop disease because of their incomplete inactivation.
Mhm.
So I also shared, and this is a mechanism for X inactivation. We're talking about the this is center of the inactivation center, X inactivation center, and that gene is quite an important gene, XISD gene. So so that's related with this one chromosome. In the one chromosome is a this chromosome we design like inactive X chromosome. So this gene in the X chromosome will be active, and the active X chromosome is not express that gene, and that gene is coding for the one quite a long RNA. So this RNA is not translated to the protein. This is regulate RNA. That coding we call this a long non-coding RNA. It's quite an important regulate material right now. So you see that in a lot of part in our genome is not coding. So how important that kind of thing. So the scientists identify they concern from very concerned from the population to general to the population. So nowadays the function of that gene is available, discovered. That is a long non-coding RNA. So they join to the regulatory of the function of the cells. So in here you see that this gene express that RNA a lot, and then interesting, they bind into some factor, and they make the coating for whole the chromosome, and this coating it recruits some kind of factor coming and making some change in the chromosome like histone deacetylation, and also the DNA methylation, and make DNA is more condensed, and that's become the bar body. So that's its function of that gene, and also the mechanism related with the X chromosome inactivation. And two mechanisms is related with this gene is downside, in fact, is methylation and histone deacetylation. Okay, now is we go. Do you have any question related with this one? It's quite clear, right? So if you have any questions, so we go to the real part. So sex linked inheritance. In the sex chromosome, we have the Y chromosome and the X chromosome. But the thing is, Y chromosome kind of very small, not very small. They have about 60 megabase. But number of genes in Y chromosome is quite very small. So mainly focus we're talking and more complex is the X chromosome linked to mutation. So we also have the same pattern with the autosome is recessive, dominant. Yes, same thing. But the pattern is quite different due to the different, like the man only 1X and the female only have 2X, and we also interfere in explanation of the disease by the X chromosome. I mean, it's random X inactivation in the women. And also we have also the same factor make difficult in the explanation of the disease related with the variable expression. Same with the last presentation we have in autosomal disease. And we also have incomplete penetrant also. So that is also making the sex-linked inheritance in higher level of the quite hard to understand. But we try to explain this, and we also have some example, and we can discuss and analyze that. So X-linked first and most common in X-linked disease is we have X-linked recessive. It's more complex compared with X-linked dominant inheritance. So it's kind of complicated. It's quite complicated. And depends on sex, affect people and genotype parent also, genotype parent. So the well-known disease, we have the hemophilia A. So this can be seen here. So that is the man is more serious than women. Only look like it's almost in a man. So that's you maybe know is another name. So this one is we continue bleeding to die if we have no any kind of medical interference, we will die because of we kind of has a mutation related with clotting. I mean, clotting factor will be not in our blood, so our blood is kind of bleeding always if we have injury. Because they're kind of dangerous disease. Another is Duchenne muscular dystrophy, like this. So the muscle is kind of not developed. So it's kind of the phenotype is kind of is not developed in the body. Question. So another and very popular in many population in the world is red gene color blindness. Red gene color blindness. Let's check. So it's kind of interesting in this disease because of in our lab we also have a small project related with identifying the mutation related with this gene in Vietnam. So somebody in our lab also working in this project, and hopefully we have some kind of finding new mutation related with this disease in the Vietnamese, and probably we can publish this one. But is we didn't deeply start yet. So this disease making the phase is not related with harmful for life, not very seriously. We just mixing to recognize some color in the environment. Like in here, we see that in here we have the very full color. We recognize normal people, and that's disease people. We had lacking the ability to recognize the red color. See, the red color cannot recognize that, so they become green. So that relate with wait, what is this? That's relate with some kind of mutation in the one gene located in the X chromosome. So I didn't make the slide for this kind of disease more clear. But that's but this protein recognize this quite related with this disease is rhodopsin and alsin protein is a kind of cell membrane protein, and then they kind of sensitive to the light, very sensitive to the light. When the light come in, and they see they like the cells channel, so they're very sensitive to the light. So if the light changes, so they open the nerve cells and open the signal cells. So if they open the signal cells, our nerve system can recognize the light very quite perfectly. And this kind of those protein kind of have some kind of this belong to the quite small protein family. So from very beginning, so that gene just have one copy, but it's lucky that is in the evolution because of need of this recognized color. So we kind of increase their increase the number of that gene, and some gene has a mutation. That's a good mutation, and they allow us can recognize different type of color. So there are two genes. There are two types of gene. Is located in the sex chromosome, X chromosome, and there we also have we also record a lot of the mutation related with those genes in X chromosome, and make their color blindness disease. So the color blindness is quite very diverse in the expression, because of some people cannot recognize the red, some people cannot recognize the gene, and the level of the recognition is also different from people to people due to the mutation in the gene. What I read is red gene is we have just one gene, and green gene is we have more than 2 genes. Some cases we have more than several genes, green gene in the X chromosome. Okay. It's quite interesting. So this is other disease. Many kind of disease relates with gene in sex chromosome, X chromosome. This is also related with the cleft palate. This one. The face we have a cleft in the face, and some baby is the face is we need some surgery to fix that. In Vietnam we have a lot of that case. And a lot of the people need to be genetic checking before they make a baby, because this also I think that is quite very popular in this disease. So the consequence is quite a big difference between female and male in X-linked recessive disease. So in female, because we have a 2 X chromosome, so the effect in the female kind of less compared with the male. So so if we have the 3 cases of the female, right? So we have a homozygote of the normal gene. In that case, we have nothing to say, because that female is no any kind of disease, she healthy. And next is she had the homozygous of the disease gene. So she will express the disease phenotype completely, right? Because of she don't have the protein, or she have the high dose of protein in any cells. And another case, and this is a more common case, is we have a carrier with a heterozygous, one normal chromosome, one normal X gene chromosome, X chromosome gene, and one is abnormal. So it's quite different from the autosomal recessive. So in autosomal recessive, in autosomal recessive, you remember is we will not develop the disease. If we develop, it's kind of quite the effect is not small, right? But in case of here is in case of X chromosome in women, so we have the 50% cell is normal, and 50% cell will express disease phenotype. But somehow the effects of whole body is kind of less. So in total, 50% of normal protein level is okay. It's quite similar in case of their autosome. No effect on my effect. But in male, it's quite different for males. So we had a homozygous of disease gene, and completely we express the disease, right? So kind of simple, no question, right? And we discussed about the frequencies of the X-linked recessive inheritance. So frequencies of disease is the most different between male and the female. I mean, if the people is have the disease in the population, have some fraction of that disease alleles in the population. So the female is much less compared with male. So for example, so hemophilia A is we have the 10,000 male, we have 1 people is affected. So Q in here is because it's any males in have that gene is will express because he don't have the compass dose. I mean, see he don't have the second option to rescue the bad gene, bad alleles. So the Q is like this number. But affects female, it should be the Q square, right? Q square. Like you see that the fraction of the people like the last time we calculate for frequencies of their homozygous. So only homozygous in female can express the disease. So this number is kind of very less, 10,000 less than male. So we have a conclusion, very easy to understand, that is X-linked disease is much more common in male than in the female. So female is very strong, right? Another one is we talk about the transmitting pattern. So this is common pedigree. So the common pedigree is the married between the normal male. Maybe the effect male is the outcome phenotype is quite terrible, so they cannot get married, for example. And carrier women marry with a normal male, and you can see that. So the female they transfer directly to the male. See, so in here we have them. All male is have the disease. Only male have the disease. So because of and the female also have the gene. Female also have the that's carrier with gene, but the effect is not high. So she don't express the expect outcome of the disease. So she just go and marry with a normal male and also keep the history of the family like this. So the disease is still coming to the male. So we can see that it's no father to son transmission. No father to son transmission, right? Because we have in the pair of the sex chromosome a female, a male. So the father just gave the son Y chromosome only. But now we're talking about a disease related with gene in the X chromosome. So father cannot the cause of the disease in the son, right? So if the son had the disease, so mom is the one is the carrier, the affected disease, and but we have some case that the effect male also is in the mating with the carrier women, and that is we have the X chromosome. But the thing is also is the father is not give any kind of X chromosome to the male, so that is also is not happen. But if the dad, the female, if the male is the effect, if had a bad gene, so on daughter, because of on she gave the X chromosome to the daughter, so on daughter will be have his bad gene. So on the his daughter is will become a carrier in 100% in the daughter will become the carrier for the bad gene. So here is also illustrate again that thing. So in here we have the affect father, and on son is a kind of safe in effect. But on daughter receive his X chromosome is error, so the daughter is the daughter will be carrier, and they will pass the bad gene to the to next generation in the son. And if mom in here is a carrier, she will transfer that one in here for this case. So transfer in this case, and she will give that bad gene to the affection. And this is a less common mating. So we have father, effect father, and also effect mother. So in daughter, so 50% effect and 50% is carrier. And son is 50% is normal and 50% effect. Oh, so this one is mostly serious because of both father and mother is effect. Sorry, his father is effect and mom is a kind of carrier because of she kind of normal in one chromosome, and X2 is a kind of a disease chromosome, for example. So in here, we can see is father to son can happen. So in here, right, father can transmit their X2. Father and son also is 50% effect. But in here is, like I mentioned, that father just gave Y chromosome, normal Y chromosome to son. So the son is affected not by the mom, but by the not by the father, but by the mother. So in X-linked recessive inheritance, we also another case make this one is more complicated to explain. That is a female with a heterozygous for recessive disease also affect. So not always we have the mild effect or no effect in heterozygous, right? So because of the, in case of hemophilia A, so about 5% of the female heterozygous will exhibit the disease phenotype. Because of what? So that's because of the X chromosome inactivation is not random, random. But random is not 50% always. Sometimes it's about maybe 90% and 10%, 90% and 10%, or 30% and 70%. If the fraction of the disease gene is become the inactive chromosome, so that disease form will be expressed more than the normal copy of gene. So that's because of the not random, so not always 50-50. Okay, so that is the reason the female is quite a heterozygous. In theory, she will not affect, but she had effect. But it's not the serious because of the some percent of her body is still normal case, normal cells. We have the case of the female who express her own disease on X chromosome. This one is what? So you have any question about this one? I forgot this one. I put this one. So sometimes in the X chromosome also we have the segment rearrangement, also make kind of disease form related with this one. It's also some part of another chromosome we jump to the X chromosome, or recombination of the X chromosome also happen. in some case. So it's make the pattern is different with the normal case. Okay, before we break, so we're talking about the X-linked dominant inheritance. It's kind of short. So in here is like I said, that is the recessive X-linked recessive inheritance is more common, and X-linked dominant is not common, less prevalent, and it's quite different with very different with the recessive case. Is in female is more common than male, twice common than female than male. So anybody can explain for this case why is in female is more common of the disease related with X-linked dominant compared with the male in the prevalence. Why female is more than male in this case? Anybody? Help. Why is female in this case, in this kind of because female have 2 X, female have 2 X, so it's more seriously. X in women is twice than male. But this in normal case, the 1 X is inactivate, right? Inactivate. So that if that gene is inactivate, so if that gene is inactivate, so that means the protein dose is also same. And just about 50%, no, about 10 or 15% is X gap. So it's not account for about 50, twice time is maybe more than male. Another idea? Yeah, correct. Because male is really suffer with the dominant disease in X chromosome link. So the male is die in development already and cannot develop to the full term. So he died already. So in the real life, that man is not available. So in that is a one is accept this explanation. Okay, good. Thank you. In here, we also have no father to son transmission. In here, you can see that in X2 is a disease copy in this chromosome. So she transferred to male, and it's male got disease like that. And also mother and transferred to daughter in this case. So daughter also expressed the disease. This and male is quite maybe it's not and die, and the number that man is not much compared with the women. So we see is only women express the disease. And because of dominant, so they also is less skipped generation. I mean, skip generation is very popular in the recessive autosomal recessive disease, right? Autosomal recessive disease. When we got the A, small a, and small a, when we can have it. But in case of the In case of the sex chromosome, because of the sex chromosome, we have only one. In the man, we have just only one. So skip generation is kind of popular in the autosomal recessive disease, but in the X-linked dominant or recessive is also expressed not skip generation. From this generation to next generation, disease people we can detect. So we have some kind of example like this. So we have the hypophosphatemic ricket disease related with some kind of protein in the cell membrane. So the kidney and somehow is kind of cannot reabsorb the phosphate anymore. So we need to reabsorb the phosphate in the kidney. So the bone in our is kind of easy, not developed well. You see a phenotype like this. The phenotype like this. The bone is not well developed like normal people. Another one is also is very serious system is this disease, incontinent pigmented type 1. So only happen in female. Maybe in male is all the male is die already. So the female is have the skin is have come like this pattern and very easy to develop the cancer. And another one is Rett syndrome. Rett syndrome related with the this gene mutation. So normally this gene is will control the condensation. Condensation. That means make some part of the chromosome is a condensation to inhibit an activation gene. But this because of this gene is mutated, so that not happen anymore. So the chromosome dose may be increased. Chromosome dose may be increased because of the gene is still expressed, not the inactivated like normal case. So it's make a lot of relate with the nerve system and also in here is the case of the Rett syndrome, very heavy. But a lot of cases in women is they have a mild effect. Is the face is kind of normal, not serious like this, but they also have the variable in expression. And this is X-linked dominant inheritance. So you see that the female will directly get the if the female is have the gene, effect mom will be transferred to both son or the daughter will affect and directly affect this. So the female right is not now is not carrier anymore. So in all the case we have no carrier. Okay. So if the affect father, they will transfer the on to the daughter only, and the son will be safe because of the son is not received the X chromosome from father. So and this is the comparison between the dominant and recessive inheritance. So let's just summarize again. Okay. Okay, we can have a break about 10 minutes and come back and we will go through on it. Today we can stop soon. We have just few slides remain, but interesting. Okay. Okay, anyone? So we continue our presentation. You come back. Okay. Thank you. The lesson today kind of quite short, but we go a lot of things about talking about a pattern. Talking about pattern is kind of if you practice with this, it's more interesting. If you have some case to specific analysis and also some case to do the research, it's more interesting compared with just we're talking about the pattern of the disease, I think. But the thing is, in here we discuss something quite very basic, It's a basic talking and basic pattern of the disease, genetic disease. Why kind of this basic recognition is kind of important? Thinking about we have some people in our society because of in Vietnam is kind of we kind of economic condition is not very good. But in the case of the another developing country, is kind of very they very care for the baby. But in our country, we can contribute to another effect, like we can contribute somehow for we can decrease the prevalence of the bad alleles making the disease in our population. That's also very great contribution for the society. For I just thinking about the example when we have somebody is we get birth, but and even they grown up a little bit in the teenager, but develop some kind of maybe quite late, but in not very early, but he developed some kind of disease in his face or his he have some symptoms, they kind of serious. And you see, is we just have the body quite complex, but somehow the phenotype may be same between disease and disease, kind of very similar, and somehow we cannot not distinguish it, right? Something we can see say that maybe he's have some fever, it's easy to eat the drug and fix this, and somehow we cannot fix, or the maybe some kind of disease. We have it in that disease in the slide too. We cannot see. We totally blind at about 30 years old, and somebody is a blindness, become a blindness completely. That is related with genetic and late onset of disease. So first we need to if that people is ask the doctor, and how you can see that the doctor can respond, because the doctor is maybe not really focused on this one, the genetic. So doctor just normally relate with a normal disorder, maybe infectious or some abnormal in the disease development is some common disease, but some rare disease he don't know. But you are the one is prepared those knowledge and some kind of technology and also some kind of skill for communication to help the people to find out what kind of problem they face up. And that is really helpful. So in the book, they also and throw some letter of the mother. You see, have the baby. That baby is suffer with some kind of disease. It's the phenotype outside kind of slightly different with a normal baby, but the inside, the biochemical and digest or some kind of the clinical trace is not normal and still recognize it. But the problem is she don't know what kind of disease, and doctor maybe also not know well. So in here we come to relate with genetic disease and careful analyze the case by case, and you may recognize the phenotype first and then check you need to discuss between the doctor, between some scientists related with genetics, and also related with medical genetics. So we can make a hypothesis like these people maybe make that kind of specific, because new disease maybe no, it's a lot of diseases that we know already. So we can discuss and we make the hypothesis, and then we need to do some tests, some molecular tests or some cell tests like RNA checking, because of the most of case related with the protein, and we need to check RNA, and then we come to check DNA, because that is look like easy, but actually it's not, because of the very rare disease. So we have no kit, we have no any kind of facility for the experiment for check that can be disease, right? So hard to test, especially maybe some chromosome or some kind of the segmental gene. That means of the big segment in the chromosome, maybe the deletion or insert or somehow translocation. Hard to detect, very hard, very difficult to detect that kind of defect, and hard to confirm your hypothesis is correct. But if you know about the disease and you know a lot of information, and that's very helpful for the patient. Some kind of disease, like I mentioned in the previous slide. So some kind of disease is they have just small effect, but the thing is they have, but all of that kind of disease will come to their cancer in a few years later. So if you know it, if you know that the disease name and you know the characteristics of disease, is easy to develop a cancer. So that patient is normally they will check cancer tumor very frequently. So if he can detect the cancer soon, so he can rescue that. That is also very valuable for this life, right? Okay, so that is quite a meeting. So we need to come across is many kind of disease pattern, and let you see that it's not very clear between the recessive or dominant, between the X chromosome or Y chromosome. Y chromosome is we talk it's quite very easy, and many cases of related variable in expression, like incomplete penetrates. So it's quite complicated and not easy to recognize. You have to develop, and me too, to develop the ability to recognize the disease definition, identification. So we also go to the final relate with the chromosome disorder is a Y-linked inheritance. So this is the most easy to recognize compared with another pattern. That is very simple to see. So we just see, if you draw the pedigree, you just see there, you can see the pattern easy, right? The pattern is directly transmit from father to son. You see, all the male is got that one. The female don't have a Y chromosome, so they don't develop the disease. So Y chromosome also is dominant, right? Because of we have no X chromosome to compensate for the male. So if he had the mutation, that's also expressed. So we don't have the skip generation from this generation to next generation, and also the downstream generation. We also have that one. And because of the Y chromosome is kind of small, not very small, but they're bigger than chromosome 22, I guess. But the number of genes in Y chromosome is number of genes in the Y chromosome is small. So we don't have much of disease in this chromosome. So the pattern is quite very simple. Father transferred to son. that is finish of this one. Nothing to say. Any question about this one? No question?
No.
Oh, no response. Okay, so we go to the next, talking about the kind of misidentification of the disease pattern. Some pattern is quite very similar with the sex relates disease, sex chromosome relates disease, but not. So in here we had a two term, is sex limit and sex influence traits. It's like this. So sex limits is occurs in only one of the sex. So that means some kind of phenotype is expressed only in male or expressed only in female. But that's not correct. It's not related with any gene in the sex chromosome. That's because of the anatomical difference between male and female. So one very easy to thinking about is the inherent problem related with uterine or the testicular defect in the female and the male respectively. So so in here is because of the maybe that gene is kind of we also have in male and female, but expressed outside is different between the male and the female, and we kind of misunderstood and misexplanation that is a sex chromosome. So, but we have a sex limit, and what is sex influence traits? So sex influence is different with sex limit. Sex influence is sex limit is only in the male or only in the female, and a sex influence is they also not relate with the gene in the sex chromosome, but because of some factor relate with the sex, so the effects may be bigger a little bit compared with another sex. But in here we have the one like here I extract this one from the book. It's not mainly due to the sex chromosome gene, but partly related with the sex. And maybe the phenotype may be elevated by the sex effect. For example, is a baldness. So the hair loss, the hair loss in the male is more serious and more common than female. Female also have their kind of baldness. You see, is the number of hair in the female is less than male. Okay, that is true, normal case. And this relate with part of sex different in hormone level. So some kind of disease and development of the disease outcome is rely on some pathway, and that pathway may be relate with some kind of sex protein, but the sex protein is located on the X chromosome, for example, maybe. So in here we also the bias is also serious in male because of the some kind of hormone in the X chromosome in male also help this one more serious. And we also have the recently we identified some gene in the autosome gene. I forgot which is, I forgot this one is a Y chromosome, but maybe this is related with the mental gene, but related with the baldness. But this one is very popular when we see that is we had a pattern is father transfer this phenotype hair loss to son, father to son transfer. But you see, is father to son is not we cannot see in the X chromosome related disease, right? X chromosome is not father to son because of father to son is we only have the Y chromosome related. X chromosome related is not father. So father just give the X chromosome to the daughter only. Okay, so Okay, so we kind of finished about talking about the nucleus genetic material related with disease. So now we come to the mitochondrial inheritance. So mitochondrial inheritance is quite a very nowadays it's quite simple, but it's very serious. Like I kind of when we open the presentation also concerned about. So because of very important for the cell survive, right? Because of energy. And so I have some kind of old information about our human mitochondria. So we have the genome size of the mitochondria. So quite similar with the plasmid in bacteria, about 16 kb. In there, the encoding for, remember we have no intron in here because this is related with the bacteria. So we have 2 ribosome RNA, and we have 22 tRNAs, and 13 polypeptide related with oxidative phosphorylation. So oxidative phosphorylation related with the fire, the sugar, and for the electron transfer, we generated energy for the cells, making ATP. So and of course, it's a number of gene and very small genome is not enough for a mitochondria can use. So normally the cells and mitochondria can absorb about 1,000 nuclear gene. The gene expressed in the nuclear and the protein product is transferred by the Golgi system to mitochondria for the work. So that is a normal happen to mitochondria. And another one is we have to see is because of there are one kind of facts about the mitochondria related disorder is the variation. I mean the disease outcome is very kind of diverse between people and people. There's one reason of that. That is, there are one cells, they have a lot of mitochondria. One cell have a lot of mitochondria. And I come it later. So I continue talking about some information about mitochondria so you can easy to amaze about that kind of effect. So the mainly is a maternal line, is not paternal line. Because of only the egg, we have a sperm and eggs making the zygotes, and in the eggs is mitochondria only in the egg, and the sperm we have it, but very less. And if we have it, that can even is also very rare. It may not happen, and very less information like that. So mitochondria from the father, it maybe is not contribute at all in the embryo development of the zygotes. So so mainly is maternal line. So that's what's mean of that. So it means if mom have some mutation related with mitochondria, so she will transfer all to for all the baby, not distinct between not different between man and daughter or son, all effect, right? And the father don't respond for this kind of, don't take any responsibility for this one. And how about the mutation rate? Mutation rate of the mitochondria. Mutation rate of mitochondria is 10 times higher than nucleus. Somebody can help me to answer, explain for this kind of thing. Why is it in the same cells, mitochondria mutation rate is higher? Okay. No. Why mitochondria had a mutation rate in the genome larger compared with the nucleus? Smaller size. Okay, very good, correct. Okay, anyone to know that. So that's correct, totally correct. So because of the, you see is in nucleus, we have a lot of gene. Maybe it's more than 20 or 30 gene. It's related with DNA error fixing. That's a preparation for the DNA. So, but in mitochondria, we don't have that protein. So we don't have mechanism, like you said, mechanism to fix the mutation in the mitochondria. That's why mitochondria is higher than a nucleus. So that's very correct. So thank you. But I didn't compare between mitochondria. Maybe now I have a new question. Is how about a comparison between the mutation rate between the bacteria outside compared with mitochondria? So I didn't ask, yes, that's not a question. So I just remind about the question to the question. So compare between the mitochondria genome and the genome of the bacteria. They were different. So that's maybe also very good. So the mutation rate, so okay. So in here, we have the term is heteroplasmy. Heteroplasmy in here. Heteroplasmy is a portion of the mutation of the mitochondria per normal in its cells will be I mean it's in the one mitochondria. How many plasmid? We have a lot of plasmid, about 100. I not remember exact number, but about 100 of the mitochondria of the right. I'm not sure about this one number. About 100 copy of the plasmid. Oh. Anyone still okay, right? So I just notification that my internet has some problem. Can you hear me well?
Yes.
Okay.
It's okay.
Yeah. So heteroplasmy. I mean, because of the high mutation in the mitochondria, and the number of mitochondria in each cells is also high, maybe 100. So I mean, in one cells, maybe some different alleles maybe not correct. Different version of the mitochondrial genome in our cell, in one cells, right? So I mean is different is we say that hetero also different. It's plasmid is a plasmid related with the genome of the mitochondria. So if we are more mutant, and as the I mean the portion of the mutation compared with the normal mitochondria, it means that ratio is high, and then we have some effect, more effect. You know, is mitochondria is very important, and something is important is the nature is not allowed to change, right? And if they change, we have problem, and that's why we have problem. So, but mutation also is always happen in mitochondria, and that's maybe the problem with the cells. We will come later in this slide also. And we have the random divide in the cell propagation. Why? What's this one? So I mean is when the cells when the cell propagation from one cell to become two cells, the mitochondria is not like that. Not like that. The mitochondria are not divide the same way like the cell, not the same time with the cell. But maybe in the cells have the 100 mitochondria, maybe 30 will come to this cell and 70 will come to another cell. That's random. We're not sure about that. And then when the cell need more energy and the mitochondria start to propagation from the some mitochondria to make a lot of mitochondria. So we have the that is explained for why the defect relate with the mutation in mitochondria will have the very high variation in effect, because the number of mitochondria from this cell to another cell, from this people to another people, is different. So I also asked myself about how about we can diagnose for that one thing. Because if you think you design a primer to PCR any kind of mutation in your mitochondria, that also makes it difficult to recognize the signal. If it's too low, actually they have, but you cannot detect that yet. So you can think about this is the effect is more serious compared in the organs need a lot of energy. For example, in the nerve system. The nerve system is used about 20% of the ATP in our body to for what? To turn back the sodium ion from inside of the cell out of the cell again, and that is make the gradient of the ion in the outside and inside of the cells for the normal function of the nerve cells. So a lot of mitochondria mutation is commonly is in form of the missense mutation, making the protein is change, mixing protein or change, or the ribosomal RNA tRNA changing. You see, it's a lot of disease in the nucleus relate with the regulatory system, right? Regulatory system. Because we have a lot of intron, we have a lot of things related with the regulate, like the enhancer, repressor, or promoter. But in the mitochondria, those things is very less, right? They maybe just have the one promoter, and then they can express a lot of protein. It's very simple this thing. So the most well-known the mutation is just a missense for the protein change in the coding of the protein in the mitochondria. But we also have another case like single base mutation. We have deletion and the duplication gene. But almost is related with the protein coding reason, not the regulatory, like in human. Okay, some kind of thing related with some kind of the example like the deafness, late onset, and some kind of Alzheimer's disease, and a lot of case related with the defect in the intellect. I mean the intelligent abilities of the people, because the nerve system is the most affected organ. And we have some kind of interesting hypothesis related with aging process, because of mitochondria in the old people, maybe they old already, and a lot of mutation in there, and mitochondria are not working well. It's not enough energy for do the work in the cells. So that means it's a mitochondrial mutation, maybe that relate with the aging process. So this is also a hypothesis. We're not sure yet. And this is a really what that disease. So I didn't, I put the name and here, that's disease. So labor optic neuropathy, this one. So relate with the mitochondria mutation. So this one is I take this one from the one paper from the Brazil, and Brazil, this is a very big family, but there's many members in this family suffer with this one. just about 30 years old, and that people will be suffer by blindness totally, cannot see, because some nerve cell in the eyes will be dead because have no we have problem with mitochondria cells. Okay, so very important. And nowadays, more and more defect related mitochondria also is a variation in the population or related with mutation in the late stage of the human life. Also, we identify. The pattern is also quite easy to recognize. So only female to the offspring. I mean, all offspring, not different between sex. Female or male is effect. When mom is the effect of people. And complete penetrate. I mean, it's, they will express because they too important. There are no anything can compensate that. But in variation, due to the number of mutation, genome, mitochondrial genome in the cells. So, any question related with this one? No. Okay, we go to the end. Thing is related with the genetic imprint. Maybe somebody in our class is also heard, and also somebody is in my group also work in this one. Even remember correct or not correct. What is genomic imprinting? And Mendel theory. So same phenotype. If a mutant alleles inherit from mom or dad, it's no different if that allele is come from mom or from dad. That Mendel say that. So there's not, but this one is not related with sex chromosome, not related with sex chromosome. But this is not always true. Many genes in human are inactivate from the specific sex, from this specific sex. In here is inactivate is we cannot detect RNA expression in those genes. And you see that in the almost the gene in our cells is we have a two copy, right? And that is the dose is 100%. And here's some gene is quite up. Some gene is quite up is maybe the body is not accept if they are high dose, and they need to suppress the dose from 100% to become 50% for normal activity. So this is a related result in the protein dose. So another one I need to mention is some gene, the specific, specific, what it means specific in here. Specific parent, that means that gene, for example, in the male and transferred to offspring, and only male in any kind of case is inhibit, for example. And another gene from mom, that gene come from mom is inhibit in any case of the species. That is I mean, there are no kind of the case like that gene is from mom. Somehow is some cases inhibit, and some cases express. It's not like that. It's very specific for the sex related with the specific gene. So right now, I don't know is this number is updated or not. So in the book, I just checked in the book. I didn't check in the database. So right now in the human, we have a 100 gene imprint. And another kind of the one characteristic of the imprinting gene is that they are located in the near together. They neighbor each other. So they occur in a cluster. Even though they're not either gene family, but they in the cluster. I mean, imprint gene, they are neighbor each other. And the mechanism of the imprint gene is related with very similar with inactivation in chromosome X, inactivation of the X chromosome. So we have also heavily methylation in the DNA, and also histone hypoacetylation. That means no acetylation, hypo, not hyper, okay? Hyper is a lot of the acetylation. Hypo is no acetylation. No acetylation in entails making the DNA wrap very tightly with the histone, and chromatin is condensation. We have the heterochromatin. So that is, we have no any kind of space for the transcription factor can enter and induce the expression of gene. So that is a mechanism. So this is a really recently, maybe just 30 years or 35 years ago. So I see some kind of book related with some pictures, some very top scientists, and they have the conference talking about this epigenetic mechanism. So this thing is very hard to identify, quite hard to identify. So my lab before I have some project, I'm not involved in that project. So somebody is suggest some kind of gene in his project, my friend. So he thinks his gene is kind of has some symptoms of imprint, and then he suggests some kind of experiment like methylation checking. So methylation checking is simple. We can do it in our lab. We just need to investigate a little money, so we can do it. But the thing is, he cannot find any kind of information. He failed in that thinking. But think about if that gene is very important. That gene is very important, and amount of protein is it should be maintained well for cell activity. So that gene is normally not imprint, and imprinting is in some corner of the activity. So very strictly about the gene dose. So we come to the very well known about the genomic imprint, one gene, one cluster imprint. And also we had some kind of disease related to this one. And here we have two genes, IGF2 and X19. IGF2 is what? I is insulin growth factor, insulin-like growth factor number 2, and in cluster with X19. So you see, is in male is X19 is also expressed, and IGF2 is not expressed. IGF2 is only expressed in the male, and not expressed in the male. And X19 is not expressed in male. So only one copy is work. So very beautiful story, right? Like this one. Only male have this one. Only male have this one, and only female have this one. So in any case. So the scientists identified a mechanism. This is a very famous, very good example of the imprint. And here is a picture. They found a mechanism. So we have a CTCF in here. So this is a zinc finger a transcription factor. So they bind to this part. Because of this one is methylation is not happening. So this is white color is a unmethylated. Unmethylated, that means is a methylate group is not binding to C base. And here the black color that means is this is methylate, fully methylate. And DMR is what's DMR? Differentially methylate reason. It means it between two set of the chromosome. So sorry, two strain of the between two chromosome in the chromosome pair, and we have a different in the methylation pattern between two. So in here, CTCF can bind in this and induce for X19 express. And also CTCF can have a protein and they inhibit enhancer. But enhancer is, this is enhancer for IGF-2. In the male, because of we had a methylation already. Methylation in the X19. So X19 in male is not expressed. So CTCF cannot bind to in here to suppress enhancer in here. So enhancer in here freely, so they can induce expression of IGF-2. So that is quite a little bit complicated, but easy to understand about genomic imprinting between male and female. They have to relate each other, and that's why they also explain why the imprint gene is normally in cluster. They need together to maybe become the cassettes for regulate for expression. Okay, now we go to real disease. This is I take from the book, and this is a disease related with genomic imprint. This is back with white man syndrome. So the symptom is we have a big belly button like this, and sometimes it's more serious. Some part of your organs in belly system will be pushed out of your belly wall, and you have maybe the very thick ear here, and one of the most impressed is a very big tongue in here. So you see it's really abnormal, right? Another one is this disease also is very risk for the increase of the kidney tumor. So if you know well about this disease, you can easily make the advice for this family. Normally, it's frequently checking the cancer or tumor checking every maybe 6 months for check it. And if the tumor occurs, so we can easy to have a therapy for that one. So very impressed, right? Like a big tongue. So now is to check you something. Okay. Now we go to the mechanism of this one. And also this is final slide for this today. So this is a cluster of that imprint gene. So we have the two parts, so the DMR1 and DMR2. So you remember this one in the middle, we have the methylation pattern in here related with CTCF like this one, right? So it's also same thing. So 20 or 30% cases, we have a paternal uniparent disomy. So this one is, next week we will learn about this one more clear. So we have the disomy. That means we also, in the pair of the chromosome, we also have the two chromosomes, maybe two chromosomes. In here we have a two chromosome. In normal cell we have a two chromosome, chromosome 11. Other thing is both chromosome 11 come from one parent, maybe come from one father or come from one mom. And in here in this disease we have those pair of chromosome 11 come from parent. Come from father. From father. Mother is don't contribute to any kind of chromosome 11. Or somehow we can lose the methylation in the DMR1. So means we lost methylation here. Actually, we have methylation here. Remember, right? This one. So in the normal case, the EGF2 in male will be active and EGF2 female will be inactive. And normally we have the one copy active only. But in effect, people, we have this one, disomy. So we have the two active copy from father, right? Father is active, mom is not active. So we will increase the dose of the protein. So maybe the increase the dose of the, because this is growth factor, right? Growth factor making big. And this one will contribute for big thing like this one, the big tongue, and we also very easy to develop the cancer because cancer is related with something is increase the growth. And 50 to 60% cases loss of methylation in the DMR number 2. This one. And, okay, that's the end of my presentation today. So any question so we can discuss? You still there? Any question? Huy, any question? Next, we will discuss about the some kind of abnormal related with the chromosome and the chromosome scale in the cells and disease related with that one. And next chapter we talk about the biochemical disease. But the thing is, any kind of disease related with the biochemical or some kind of small scale or large scale mutation. But I also talked in the beginning of the chapter. So we have a chapter, but they are not really I mean, it's a distinct chapter. Okay. So about the, I mean, any kind of the mutation also, if they have harmful, they also lead into the biochemical usage because biochemical activity making the energy running in the cells, make the cell work. So the core is the same, but in the book and the chapter book, they try for you to make some definition in any case and for easy to recognize that's what they want. To analyze about the disease, you have to combine all of them together, like small scale mutation, large scale mutation, and that's make some kind of thing related with and a pattern, prediction for the pattern. There is autosome or the X-linked chromosome to help you to can predict the risk for the next generation, and also easy to diagnosis for name the disease of what kind of disease, and from that we can have their treatment, or somehow is assist for a very valuable advice for assistance for the affected people can have a better life. But at least they can somehow they can survive happy and they not depressed because the situation, because we cannot change, right? Somebody is have a big tongue or somebody is a blindness. Somehow we cannot rescue that. This already happened. Somehow we can do it better. This some chapter here we talk just about a rare disease because that is a rare mutation. And we have one chapter talking about the multifactorial multigenic disease. I mean, it's one disease related with many genes. So that's a common disease. That's more, you can see it's more common like diabetes or cancer or something. Okay, so so when did you have midterm? Can you turn on your mic and discuss a little bit? When your midterm? I forgot it. 20, right?
I think it's
From 20?
yeah, from 20.
20. So we can meet about 2 weeks later. So I will upload the slide. I will upload the slide this week, and we can discuss one more week, and we will prepare for the midterm. I still don't have any idea for midterm yet because of this. It's totally different with the last year. So question also changed, I think. Maybe very short question, but I'm also scared for short question. But the score is just 25%, so that's fine. Okay, if you have any questions, yeah. If you have any questions, so we can stop here. So have a good day. Have a good day.
Have a good day.
Yes. Goodbye. Thank you. Goodbye.
Bye.
Thank you.